I was talking about the WINTHER trial, which is the first trial run by the WIN Consortium, and it is really a cutting-edge precision medicine trial. It was the first ever trial that incorporated not just genomics but also transcriptomics in order to match patients with drugs. So I think that the WINTHER trial gave us extremely important information. So when the WINTHER trial was created in about 2012, we had genomics and really nothing beyond that in the clinic. So our first question was, will transcriptomics help us increase the number of patients that can be matched? Transcriptomic is RNA expression, so the genes are turned into RNA, and it makes sense that the RNA would be important mediators of what actually happens in the tumor. And so the first and maybe most important thing that the WINTHER trial showed us was that the RNA data was valuable. So we do have other findings in addition to that. The other finding was that it's not RNA data just from the tumor itself. You have to compare the tumor to the normal tissue because the normal tissue has tremendous variability. So if you look only at the tumor, you're going to get a false impression of the RNA. You have to look at the difference between the tumor and the normal in order for that RNA data to be valuable. If you don't do that, the RNA data simply isn't going to be valuable. So that is another important finding from the trial. A third important finding from the trial was that matching patients to drugs is not a yes or no. The degree of matching makes a difference. So we developed a matching score, which looked at the degree of matching. And that means that if patients had a higher degree of matches, so if they had a lot of alterations, let's say they had 10 alterations and we matched one of them, that would be a 10% match. But if they had one alteration and we matched that, that's a 100% match. And so one of the questions is, does that make a difference? And the answer is absolutely, that makes a difference. A 10% match resulted in less improvement than a 100% match. So we now know that it's not just match yes or no, it's the degree of matching, and we need to optimize the degree of matching in every patient. There were several things I think that stood out for me. First of all, I thought Jennifer Doudna's lecture on CRISPR not only was extraordinarily clear and made a very difficult subject simple, but it is apparent that the power of this technology that she helped discover may revolutionize several different diseases. To be able to take a gene editing tool like CRISPR, give one treatment, and correct a mistake in patients' cells, and she gave the example of sickle cell anemia. It almost boggles the mind, but it's conceivable theoretically that this could cure sickle cell anemia with one treatment and do so permanently. Now, of course, we need to do the clinical trials and there may be side effects that we're not aware of, but this is absolutely transformative for some of the diseases that exist today.